Okay guys, welcome. So I'm going to make this short video to show you guys a few things and answer some common questions that have come up from modules 1 and 2. And this is based on what you guys submitted to me in that clarifying questions assignment. So the things I'm going to cover are some questions people had about polymerization. A lot of people had questions about that first case study. And then in looking through people's references, there's still some confusion about what's a primary research article versus a review article versus a popular press article. So I'm going to go through that and how you can find research articles to add and improve your drafts. So the learning objectives for this video, you're going to describe the steps of polymerization and analyze the benefits of hierarchical assembly. We're going to assess the effect of altering just single amino acids on the overall polypeptide structure. We're going to distinguish between review articles, primary research articles, press articles, and finally, I'll show you guys how to find some quality primary research articles. So first, let's start with that polymerization question. So there is a natural hierarchy to the cell, and we covered that in this first module. So we started talking about small organic molecules, things like monosaccharides and amino acids and nucleotides, and how they come together to form macromolecules, which make up the supramolecular structure, so things like membranes and cytoskeletal elements and our chromosomes, and then eventually those make up our organelles, our mitochondria, our nucleus, etc., and all of that come together in our cells. And so macromolecules are made from the polymerization, from the putting together of smaller organic molecules into those long chains. And this is going to be true again mainly for our polysaccharides, our proteins, and our nucleic acids. And so we start with our inorganic precursors. We make those small organic molecules like our monosaccharides, our amino acids, and our nucleotides, and they come in form of macromolecules. Now keep in mind that lipids are that unique thing. They don't have that same polymerization that you're going to see with the other macromolecules. But the benefit for all of them is that those smaller building blocks, those monomers, allow for similar machinery to be used. So we can make polysaccharides in different places using those same enzymes, for example, and then ease of transport. So it's much easier to move around small organic molecules than it is to move around a large macromolecule. And we have three major biological polymers. Again, we've got proteins, nucleic acids, and polysaccharides. And again, lipids are macromolecules, but they don't follow that same pattern of monomer to polymer. So our principles of polymerization, we're going to make them through a stepwise addition of similar monomers. We're going to have to remove water, so we call this a condensation reaction. The monomer first needs to be activated in order to form that chain, and the activation usually couples a monomer to a carrier. Usually this requires energy, most of the time in the form of ATP, and there is an inherent directionality, and this becomes important, especially as we talk about what this information is used for, what these macromolecules are used for. They carry information. So in the same way that when we read a book, when we read language, we go from left to right in English, and the direction of those letters is important for the meaning. The same is true in biology. The direction of our nucleotides, the direction of amino acids, the direction of sugars, even that direction for how they're put together, that can give us meaning. So how do we do that? Again, we're going to activate that monomer, usually with ATP. We're going to have a condensation reaction. See, we're going to lose water. We'll lose one of those carrier molecules here. And we'll continue that polymerization. So we're going to keep adding activated monomers through condensation reactions. Now let's talk briefly about the first case study. So this was designed to be challenging. I know people, a lot of people ask, like, where do I find this information? It's not directly in your textbook. I wanted you guys to really think about it. And I wanted you to think mainly about side chain chemistry. So the first part asks you, or first introduces you to the problem that we're working with. And this is Alzheimer's disease. And there is a genetic form of Alzheimer's that's caused by mutations. And this is true. So it's caused by mutations in the gene sequence, and one of them is an isoleucine to a serine. And what I want you to think about with that, and why I asked you to draw them, is because this is going to change the secondary structure. The isoleucine shown here, remember, this is our backbone for our amino acid, and then shown here is our R group, our side chain. And for isoleucine, this is a very non-polar amino acid, right? You can see just carbon, hydrogens, right? Nothing fancy happening here. However, when we switch to a serine, all of a sudden we've introduced this OH group, and this is going to make this amino acid a polar amino acid. And now not only have we changed that one amino acid, but we've changed the chemistry of that region. So now we can form hydrogen bonds because we've got this OH group. It's polar, so it's going to want to embed itself into aqueous environments as opposed to inside membranes, right? So usually this is supposed to be in the hydrophobic region. It's no longer going to be able to be there. It's going to want to be in the hydrophilic region of a membrane, for example, so interacting with those head groups or push it out fully into the intracellular or extracellular side. And using that, I want you to go through and think about these other side chains and what is the chemistry of that side chain. And so to do this, you got to think about what is happening with those different R groups, right? Are they charged? Are they polar? Are they these special cases, right? Or are they nonpolar? So you got to think, okay, sulfur, we're going to now introduce some disulfide bridges, some sulfur chemistry. Rings, we're going to be able to do ring stacking. And then you also want to think about the space that they take up. So if we're changing from, let's say, a glycine, which is our smallest amino acid, to something like a tryptophan, which is really, really big, now that can't be as tightly packed in a globular protein, for example, right? Because it just takes up a lot more space. So you want to think about what kinds of interactions are going to change, both with charge, polarity, and with space. Okay. And then part 2 was really the extension part. So part 1, I expected people to figure out based on what they've been reading about amino acids, and part 2 is the extension. So this brings up something called mutation analysis. And what that means is we're going to selectively delete each helix. So we're told that presenilin is what cuts this APP, this amyloid precursor protein. It also is frequently mutated in genetic forms of Alzheimer's disease. And so to figure out where that cleavage capacity is, where is the enzyme part of presenilin, and there are 4 helices, we're going to selectively delete each of them. So we're going to delete one, we're going to delete the second one, we'll delete the third one, we'll delete the fourth one. And so we'll end up with 4 mutant enzymes. And then we'll mix each of them with APP, and we'll measure the amount of APP that's been cut at the end. And we're going to do that, we'll find out which helix has the cutting capability. And then that last part, once we've identified the motif that cuts APP, we can then figure out what amino acid is by changing each amino acid to glycine. And again, glycine is that simple amino acid. The most important part for this section was for you to identify what is our independent variable and what's our dependent variable. The independent one is, again, the thing that we're changing, we're altering. So in this case, it's the helices or the mutants. So we're changing those around. And the dependent variable is what we measure. So we're measuring the amount of APP that's cut. And again, that depends on our independent variable, on our enzyme, on our cleavage. Okay, I hope that cleared that up a little bit. And again, case studies are ways for me to see how you're thinking about things and to clue you in onto how I want you to expand and push past what we cover in the direct material and how you should apply it to other material. Okay. Finally, let's talk about a primary research article. So a primary research article describes an author's research question and the experiments that they performed to answer that question. And so it's going to show the results of those experiments as tables, as graphs, and then explain what those results mean. And this is compared to a review article that's just going to summarize the work of like a bunch of different groups. And it doesn't present original data. So all the data has come from other sources and they're summarizing it. Both of them appear in academic journals, and this is where it gets tricky. So this is compared to press articles, which appear on news sites, society pages, etc. So how do you find primary research articles? Your best bet is something like Google Scholar. You can also use a website called PubMed, which is the website that I generally use, but most students prefer to use Google Scholar. Totally fine. What you want to do is you want to open the article and see, does it have graphs? Does it have methods? And as a hint, reviews are often labeled as a review or in review. Okay, so what I want to do is briefly show you how we do this, how this works. I'm going to share my web browser here. Okay, great. So if this is what you type into Google, you go Google Scholar, you're going to click on Google Scholar, and now you've come to a different page. Okay, so you can see some of the things I've recently looked at, but let's just look at Alzheimer's. Let's keep with COVID. COVID, actually, I lied. Let's go back to Alzheimer's, because COVID's not going to have a ton of research. Alzheimer's disease and depression. Let's see, and diabetes. Okay. So this is going to give us all sorts of things. Okay. Now, I want to keep it recent because that's just how I work. And let's take a look at the first article that pops up. Okay, the first article that pops up, connecting Alzheimer's disease to diabetes, underlying mechanisms and potential therapeutic targets. So you can click on it and you want to first scroll down and see, is the whole article accessible? If it's not, sometimes you can log in with your Brave ID and it will pop up. Otherwise, you want to look at it. Now, just skimming this, we have an introduction, and now it's going to start talking about some other things, insulin signaling pathways, insulin signaling. What this is showing me is that this is a review. So all these pretty figures that they're making here, they're just summarizing what other people have found, right? I don't see any method section. I don't see any graphs of original data. So that means that this is a review. Now, reviews aren't bad. Reviews are actually good. What you can do, let me go forward to our review here that we showed. What you can do with your review article as it loads is you can scroll all the way down to the reference section. And the reference section will tell you some of the things that they read that maybe helped them, right? So we can click on this random one on Google Scholar, click on it again, see if it loads. Press. Differential regulation of adipocyte PGE3 beta in distinct membrane compartments by insulin and the beta adrenergic receptor agonist. This sounds great, right? So we read the abstract, introductions, got some complicated stuff. Oh, look at here. Materials and methods. They're doing experiments. Science is happening. That means primary research article, right? They're doing things with mice. They're doing things with cells. They're not just summarizing stuff. And now we want to check, oh, they've got figures. They've got graphs, right? They've got more graphs. This is a primary research article. So you want to do that for your specific topic. So again, you can go to reviews and then use their references to help you. Okay, I hope that this was helpful for everyone. We went through a few key topics. The last thing I want to just commend you guys on is the discussion that you guys had for Module 2. I thought it was really interesting and you guys brought up some really great points. So keep going with that. And as always, I'm around if you have any other questions.